morning, good evening, good afternoon, everyone. The title of my talk is Characterizing the Boundary Between Life and Death Through Whole Genome Transplantation. Whole genome transplantation is the last step that enabled the construction of JCVI synthetic cells. The procedure takes the genome that is synthesized and assembled in yeast and integrated with Mycoplasma capricolum as recipient cells, where the existing cellular machinery is going to boot up the synthetic genome and create the synthetic cell. And these are the people who originally developed and implemented whole genome transplantation that actually enable the construction of the minimal cell. And here is the high-level protocol of whole genome transplantation, which was originally published in 2007, where you start with two components. One is the competent cell, which is Mycoplasma capricolum, and you also have the donor DNA. In the results that I'm going to show you guys, and also the original paper, the DNA used was Mycoplasma mycoides donor DNA. And what we do after we have the two components is we mix them together and add PEG. And we think what PEG does is to fuse the cells to enable the physical transfer of the DNA into the cell. And the last step is the recovery and selection, where we use a tetracycline, which is the antibiotic resistance marker in the donor genome that is not present in the recipient genome. And yeah, we'll talk through this protocol in more details afterwards. You may think that whole genome transplantation just looks like a transformation. And at this high level of protocol, yes, it does look very similar. However, whole genome transplantation is fundamentally different from transformation. Based on my understanding, whole genome transplantation is the only protocol so far that combines two components that are necessary and sufficient to create a dividing prokaryote cell. And in contrast to transformation, where the plasmids or DNA fragments are not a necessary component, here we're transplanting the entire genome that is a necessary component to create a cell. So now we have this extremely powerful and unique tool to integrate two components of the cellular machine. The natural next question is, how much can we break the cell and still be able to put it back? And going one step even further is, can we kill the cells, meaning that we make them non-dividable and still be able to revive them with whole genome transplantation? And if so, we essentially can demonstrate the creation of living matter from non-living matters. And that's my title of, characterizing the boundary between life and death. And although it sounds exciting, right? And this slide is the summary of the results that I have collected so far during the 4 months that I've been at JCVI. What I found is that if we kill all the cells, we cannot create a transplant. And if you're up for a failure story, and here we go. How we break the cells is we treat the recipient cells with mitomycin C, abbreviated as MMC, that can cross-link the G and Cs in the genome, basically disabling the genome. And the assumption is that the transcription and translational machinery in the cell is still active to read and boot up a new donor genome that we supplement the recipient cells with. And so the entire procedure is the same as the original protocol, except for we kill the cells first with mitomycin C. And this is the some more details of how we did it. We basically add mitomycin C at various concentrations and treat the cells from 1 hour to 4 hours at either 30 degrees or 37 degrees, and then quantifying how many cells that are still alive by counting how many CFUs are in the on the agar plate. And here's the result. So x-axis, we have the hours of treatment. Y-axis, we have the mitomycin C concentration. And at the top, we have dead cells. So indeed, if we increase the mitomycin C concentration enough, we do kill everything. And the key here is to see if we go through the entire transplantation protocol, whether we'll be able to see transplants, especially in the dead zone right here. And I'm going to walk through the rest of the whole genome transplantation protocol in more detail. So basically, treat the cells with calcium chloride to mask the negative charge on the cell so that it can attach to the negative charged DNA. And this is very similar to a E. coli transformation where we put them on ice for 30 minutes. And the reason why there's light, you might be wondering, is because mitomycin C is very sensitive to light. So we want to be able to get rid of all the residual mitomycin C so that it does not impact the donor genome that we supplement the reaction with. And then we mix the competent cell with the donor genome, and put the entire reaction at 30 degrees for 90 minutes. Here again, we're using light. And after 90 minutes, we'll be able to plate the cells. And here we plate the cells on 2 types of plates. One is no selection and one is with selection. And the goal is that in the no selection plate, and the number of cells in the no selection plate and the number of colony forming unit in the no selection plate and the colony forming unit in the with selection plate will be equal, so that that's an indication that all the recipient cells were dead and all the dividing cells or all the CFUs that we observe are transplants. However, this is the results after we go through the whole genome transplantation. What happens is, for any mitomycin C concentration that is at or above 20 nanogram per mil, the cell, there's no transplants. So we weren't able to show any transplantation that happened when all the cells were dead. However, here's some silver lining. If we plot the number of live cells and the number of transplants that we have across different mitomycin C concentrations, we can see that although both of them are decreasing with increasing concentration of mitomycin C, the rate of a decrease for transplants is much lower than the rate for live cells. And if we divide the two, we do see a very obvious decrease of the ratio. And as you recall, a ratio of 1 is what we're shooting for. So if we can extend this curve and make transplantation work for mitomycin C concentration that's higher than 20, we should be able to get to where we want it to be. And right now, the lowest ratio that we're getting is a ratio of 5. And another silver lining is that statistically, it is extremely likely that we're reviving dead cells. And this is very preliminary statistical analysis, where the basic question we're asking is, what is the probability that the transplants actually come from live cells? And if we calculate, first of all, the basic probability of a live cell forming a transplant based on transplantation reactions without mitomycin C treatment, where we usually start with 10 to the 10 live cells, and we usually get in the order of 100 transplants. So that gives us the probability of one live cell forming one transplant equals to 10 to the negative 8. And if we assume the probability of forming a transplant remains constant after we use mitomycin C, however, this is a big assumption here that we can't really justify yet. But if we just assume that, the likelihood of, for example, creating at least 14 transplant per 70 live cells, and this is the result that we got for the best results that we've got so far. And the probability of this happening, assuming that, probability of this happening only from transplantation in live cells is practically 0. Using Excel, Excel basically put out 0, and the smallest number that's allowed in Excel is 2 to the negative 208, which is practically 0. So then you might ask the question of, if it's so likely that the cells come from dead cells, then why aren't we seeing any transplants when the mitomycin C concentration is equals or greater than 20? Two big possible reasons. One is mitomycin C might be also disrupting rRNA and other cellular machineries. And also, it's possible that the donor DNA is cross-linked by residual mitomycin C. And so the next thing to try are, try different antibiotics that might have a cleaner reaction with the genome, and also perhaps wash the cells even more, in addition to using light to get rid of mitomycin C even more. And the third is, we still don't fully understand how whole genome transplantation works, so full further elucidation of the mechanism would be super helpful as well. So, there's still hope, and thank you for your attention, and thank you for all these people who have been instrumental to the work that I've done so far, and thank you very much. I'll take any question you may have.
This is James Dobbins-Beck from Birmingham, Alabama. Are you doing the PEG treatment before or after the MMC?
After.
After. Do you think that it would help if you did the PEG treatment and then washed to try to remove the MMC, the residual MMC from the inside of the cell?
Interesting. Could you elaborate a little more why that would be more
well, it's a
ideal?
general assumption that PEG, or polyethylene glycol, pokes holes in the membrane, right?
Mm-hmm.
I'm not sure exactly the mechanism how MMC enters the cell, but if it's entering the cell and being sequestered somehow,
Interesting.
then poking a hole in the membrane and then doing the wash step might eliminate any residual MMC inside of the membrane.
Interesting. So you're
And
suggesting a deep wash of the cell to
Yeah,
get rid of even internal mitomycin C that might be floating around.
right.
It's interesting.
From looking at your results, that's kind of what it looks like to me, is that there's residual. I mean, that's where I'd lean first, right? Is that there's residual MMC in the cell. So it's just worth a try.
Yeah, thank you for the suggestion.
James, one problem with doing the PEG before the mitomycin C is we grow the cells, we grow the competent cells with mitomycin C. And I don't know that we can grow cells with 10% PEG.
Right. Well, maybe a different antibiotic that's more labeled to light or more intense light treatment might work.
Just to jump in, I'm actually, if you guys can hear me, for a separate project, I'm going to be trying to grow Syn3.
Who is this?
PEG. This is Isaac from Dresden.
Okay, Isaac, yes.
For separate reasons, I'm going to be trying to grow Syn3 with PEG in the coming weeks, just as a media supplement for potential future experiments. So I can report back on if that hinders / how much that hinders Syn3 growth, just to get a sense of how harmful that might be to the cells.
I have no idea. And I would suspect that depending on the concentration, depending on even the molecular weight of the PEG, it's going to have different impact on the cell. And our current hypothesis of how PEG is facilitating whole genome transplantation is that it is actually fusing multiple cells together into syncytias. And so if that's the case, I did do an experiment where I quantified the number of colony forming units after PEG treatment and compared that with CFU before PEG treatment. And the reduction is around 30 to 70 folds. So it does seem like after PEG treatment, there were a significant amount of cells that were dead. However, I have not tried after reducing the concentration of PEG, whether there will be more cells that form again, just because the syncytia will break apart and form individual cells again, and whether they're gonna they're gonna revive basically from or emerge from the syncytia again, if that's helpful.
Okay.
For me,
Thank you.
Thank you.
Can I
Go ahead,
a quick
Olivia.
question have you tried electroporation? Just a guessing. I mean, it's my PEG, some cells get some toxicity. So if you have only a physical capability to disturb the membrane and then facilitate, I'm just wondering. Due to the size, I also thought about the gene gun or something, but anyway. You probably thought about electroporation or not?
I think Nasira is on the call, and John, you probably know.
Carol may still be on the call. Carol tried everything. We even bought a gene gun for this purpose, Olivia, and that didn't do us any good. And we also, I'm sure, tried electroporation. So this was 2 brutal years of Carol Lartigue's life to come up with this protocol. But to my knowledge, she tried everything we could possibly imagine.
Yeah, I would imagine so, yeah.
But I appreciate the suggestion.
Well, just guessing.
Anything else for Feilun? Bogomil, do you have a question, or are you just waiting in the wings? Since you're muted, it's hard to tell. Okay, Feilun. Nice
Thank
job.
you all.
Really nice job.
Thanks, John.
And it'll be interesting to hear what Isaac tells us in a few weeks.
For sure.